title on the screen already that this is an interesting, you have an interesting period in store. I'm Rush Holt. I'm one of the co-chairs of the Biomedical Research Caucus. And just to put it in perspective and remind some of you, the whole point of the Biomedical Research Caucus is to remind ourselves and remind the congressional organization of the need to support research in the health and biosciences, particularly through NIH. Americans value the fruits of research. Obviously, we're living longer, we're living better, and you can talk about the same thing in telecommunications and transportation and energy. There have been real advances that are directly traceable to federal investment in research. But the public, although they love the fruits of research, don't have a clue about how it is done and how it is sustained and how it will be fostered for the future. That's why we're here. You've got a really interesting case study of research that, you know, I'm sure there were times when someone might have said, what Bonnie Bassler is doing isn't really going to keep us healthier. This is, this is her sandbox where she's doing some, kind of nifty stuff. Well, indeed, you will hear that this will, perhaps in short order, lead to healthier people, better quality of life. But also, what she contributes to the research infrastructure will have payoffs in unpredictable, but I'm sure very real and very valuable ways. Bonnie Bassler is a professor of molecular biology at Princeton University. I'm pleased to say I represent her in Congress and a number of her colleagues. She developed a whole line of research based on the question of, well, how do cells talk to each other? How do they communicate? How do they even know that there's another cell nearby, let alone know what that other cell is doing and what they should be doing in response? And this has led not only to a MacArthur grant and admission to the National Academy of Sciences, but a very productive way at looking at cells, looking at organisms, looking at societies. So in fact, network theory or population biology, like how schools of fish all know to turn on a dime and move someplace else, or how our electrical grid with its many nodes can communicate with each other. Those things are a lot more related than people realize. And so when you hear people disparaging NIH grant proposals because they have this name, this title of this $1 million proposal or whatever it is, that doesn't seem to have any bearing on your quality of life or your constituents' quality of life, think again. What we want to do is maintain the research enterprise and the peer review, not the congressional review, but the peer review that allows this to move forward with quality. So one of the things we have to resist is substituting our own congressional judgment for the professional judgment of what is research of good quality. What is maybe someday going to lead to the kinds of things that we want to spend our tax dollars to help. So It's been a pleasure to know Bonnie Bassler over the years. She has a reputation for being one of the liveliest teachers and professors at the university, and also one of the most creative and inspiring researchers. So delighted to have Bonnie here. Thanks a lot for doing this. And by the way, I mean, you're here for a reason. You represent your member. Take this back to them and take the lesson back to them, please. Thanks.
So thanks, Dr. Holt. So I'm, of course, like the luckiest citizen in the U.S. because my congressman is a scientist, right? And then he's really just this local guy. And so we've known each other for almost 20 years, and we see each other around Princeton. And so it's really fun for me to get to come to his turf, right, and actually tell a little bit more about what we do back at home. And so I was going to give you just one story today, which is about how bacteria talk to each other. And so my only goal during these next few minutes is to try to convince you that they can talk. And then I have a secondary goal, which is to try to convince you that they're multilingual. And then if I manage those two things, the third thing that I really would like you to take home is the idea that learning that bacteria can talk to each other, learning about how they do it, actually has incredible practical ramifications for food, health, energy, the environment. And so that we weren't just playing in the sandbox, we actually had think that we've learned something fundamental about how life started and also about how to make new therapeutics. And so I know that it's been a while since you guys have been out of school, so I want to remind you what I work on. So I work on bacteria, and this would be a cartoon of a typical bacteria like an E. coli that's in your gut, right? And so they all look pretty much like this. They're single cells, and they're covered with the membrane, and so that's like their skin. So it keeps the outside out and the inside in, right? And then on the inside, they have a cytoplasm. That's the goop that all the biomolecules roll around in. And then what's special about bacteria is that they only have one piece of DNA. So they have one chromosome that encodes all of their genetic information. So they only have a few thousand genes, so only a few thousand bits of information that gives them all of their physical features and then also gives them all of their behaviors, right? So you have lots of genes, you're a sophisticated organism, and you have lots of interesting behaviors, right? So bacteria are very primitive. We know that they were the first life forms on Earth, so they've been on Earth for 4 billion years. And we know that every other life form evolved from bacteria. So the secrets that they figured out early on, you can imagine, came up through higher organisms. And so scientists have known about bacteria for 400 years, and they've studied them for that long. And mostly what they've done is sort of look at them under a microscope. And if you watch a bacterium, what you'll see that it does is it consumes nutrients from the environment, it grows to twice its size, it doubles its DNA and puts one into each end, and then cuts itself down the middle. So one cell becomes 2, becomes 4, and so on and so on. So kind of a boring, mundane life, just eating and replicating. And so for 380 of the years that scientists have known about bacteria, because of this sort of simple-looking life, right, they've always thought that bacteria are these asocial reclusive organisms. And so what I mean by that is once the bacteria divide in half, they have no knowledge, if you will, of their sister cells. They simply all go off and act as individuals. But of course, you guys have heard of bacteria. They have us on our knees, right? They're killing us. What we don't hear about so much is that they also keep us alive. And so the question that my lab had is how can bacteria, the smallest, most tiny organisms on this earth, be so powerful if they simply act as individuals and do their own thing? Right? How can they actually have such an amazing effect on the earth? And so that's what we want to investigate. But I want to make sure that you have a even better idea of your interaction with bacteria before we go further. So just to give you this is the Bassler perspective of the world. This is my idea of a human being. So I've made this man out of these circles, and I just use an algorithm to make each circle represent some number of cells. So we know how many cells there are in the human body. There's a few trillion cells. That's how many cells it takes to make you, all your organs, and give you all your features. So you're a few trillion cells. It turns out that every instant of your life, you have 10 times more bacterial cells in you or on you than human cells. So at best, you're 10% human. You're actually 90% bacteria, right? And so of course, this is the 21st century, so we know it's more than the cells these days. It's all about genetic information. And of course, we have the human genome. So I've remade my man out of the A's, T's, G's, and C's that are the nucleotide codons of the famous double helix of the DNA. We know exactly how many genes there are in human beings. There's about 25,000 genes. So that's how many bits of information it takes to make a human being. It turns out that the bacteria are contributing 100 times more genes to you than your own genome. So you can decide which of these metrics you like. At best, you're 10% human. More likely, you're really only 1% human. The other 99% of you is bacteria. And these bacteria are not passive riders. First of all, they cover you in an invisible body armor that we call a biofilm that keeps your body from being hurt by the environment. And all of these genes that they have in you, mostly in your gut and on you, right? All of these genes are genes that you don't have. So those genes supply you with processes that you don't encode. So you would not be alive without this repertoire of bacteria living with you. We know now that there would be no plant, no insect, no animal, no human being unless you live in this lifelong partnership with these beneficial bacteria that give you life. So the problem, of course, is that bacteria only get bad press, right? So we hear about bacteria in the newspaper when they do terrible things. So here's a couple pictures taken under a microscope of bacteria that you've undoubtedly heard of, right? And so we know that bacteria that live out in nature, that there are many bacteria that have no business ever being in or on you. And if they get in or on you, they can make you sick. But what I hope I've convinced you of is that you also have this amazing consortium of bacteria that's living with you starting from the moment you are born, that's keeping you alive. And they do things like digest your food, make your vitamins, they educate your immune system. They do all of these magical things that are essential for life. And so we now know that bacteria sort of have two personalities, these bad personalities and then these personalities that we're starting to learn about that are essential. And so once you understand that, then the sort of deeper question that I want to talk to you about today is whether you're thinking about the terrible things or the good things that they're doing. The question is, how do they get any bang for their buck, right? So bacteria are so small, you can't even see one without a microscope. And so if it's really true that they grow and divide and each goes about doing its own business with no knowledge of its sister cells, how can it be that they can either sustain our life or that they can kill us? And so what I want to try to tell you is that this way of thinking about bacteria is not the way bacteria live. What they do is that they talk to each other with a chemical language, they count their numbers, they recognize when they're alone and when they're in communities, and then they carry out tasks in synchrony that allow them to do these things that they could never accomplish if they simply acted as individuals because they're too small. And so that's what I'm going to try to show you, is that bacteria act as multicellular organisms, and they actually invented, 4 billion years ago, multicellularity. And so I wanted to give you an idea of how this started, because this idea of bacteria carrying out group behaviors came from a ridiculous, crazy bacterium named Vibrio harveyi that's bioluminescent and lives in the ocean. And so what you're looking on this slide is, well, obvious propaganda from my lab, but you're looking at, this is just a flask of Vibrio harveyi cells. These are some Petri plates that we put the cells on, and then we were just drawing with Q-tips, right? And then we turned the lights off in the room and took the picture. And so what you're looking at is the light that these bacteria make naturally in the ocean. So this is bioluminescence, like fireflies make light, except it's in the ocean. And so what we noticed about this bacteria, right, which has never hurt anything, right? It's just sort of a random bacteria from the ocean. But what we noticed is that it didn't always make light. It only made light at high cell number. So what I mean by that is if we put the cells and made them dilute, they made no light. But when the cells grew and divided to a certain number, all the bacteria turned on light in unison. And so the question that we wondered is, how does this bacterium understand when it's alone, understand when it's in a group, and then turn on bioluminescence? And what was so cool about bioluminescence is that you could just see it with your eyes, right? Bacteria are invisible. So if they're working alone or working in a group, how would you know? You can't see them. You can't see what they do. But what was so nice about bioluminescence was that it was a visible readout of this invisible world, and we could see that the cells did it in synchrony. So we thought, if we study how this happens, even though this is a harmless bacterium, if we study how they regulate bioluminescence, maybe we can understand how they know when they're alone or they're together. And so what we did by studying this bacterium that's harmless is we found out that the bacteria talk to each other. And so the way it works is as follows. This is supposed to be a cartoon now of my Vibrio harveyi cell. So when the cell is alone, it wants to carry out the behaviors that are good for being an individual. So it's doing all kinds of individual tasks, but among those things, it's making and releasing a small molecule that I have as these red triangles that you can think of like a hormone or a pheromone, and we call it an autoinducer. And so what happens is when the bacteria are alone, I mean, they live in an enormous world. They make and release this molecule, and it just floats away, so they can't perceive it. So when they don't perceive that that molecule is present, that tells them you are an individual, carry out the program of behaviors that are good for being an individual. And that means, in Vibrio harveyi's case, don't make light. But then as the cells grow and divide, since all of the bacteria are participating in making and releasing this hormone, this autoinducer, the amount of this extracellular chemical increases in proportion to cell number. And when that molecule hits a certain threshold level, the bacteria detect it, it tells them they have neighbors, and all of the bacteria turn on bioluminescence together. So the way the cells understand when they're alone or when they're in a community is by measuring this extracellular molecule and using it as a proxy for cell number, right? So they believe, if you will, if they see a lot of that molecule, that must mean that there's neighbors around. And so we can trick them. We can just take that molecule, squirt it onto low cell density cells, and they turn on light, right? So they're using the molecule as a chemical word to talk to each other and tell them they're in the neighborhood and they should all do something together. And so once we got that far, we started to bring the tools of molecular biology to this to understand how does this work. And what we found was that so this now is supposed to be my bacterial cell, is that the Vibrio harveyi has an enzyme that makes that hormone molecule, and that molecule diffuses out of the cells, right? So the more cells there are, the more that stuff there is. And then there's another protein. There's a receptor sitting on the cell surface. This is exactly like the hormone receptors that are on your cell surfaces that detect hormones that are floating around. So there's an enzyme that makes this signal molecule, the autoinducer. There's a partner receptor sitting on the surface, and when the autoinducer reaches this critical level, the receptor can find it, and it binds into a pocket, like a lock and key, and that sends information into the cell to tell them to turn on the genes that make the enzymes that make light. So what happens, right, is that only when there's lots of cells around, when this molecule is there, does the signal get sent internally to turn on bioluminescence. Right, so it's a really simple circuit. The more cells there are, the more of this stuff there is, and then together the cells turn on light. And why that is interesting is that because we initially found this in this bioluminescent bacteria because we could just see the bioluminescence that happened at high cell number, but then we started to think, why should this only be this one bacterium? So then we just started looking for these genes in other bacteria, and in the past 20 years, we have found hundreds and hundreds and hundreds of species of bacteria that all have an enzyme that makes a signal molecule. There's always a partner receptor, and then together these complexes turn on all kinds of genes that bacteria want to express when they're in a community and not when they're alone. And so now we have a fancy name for this. You guys are in Congress. We call it quorum sensing. The bacteria vote with these chemical votes. The vote gets tallied, and then everybody goes along with the decision. And so what's important is that it's not about bioluminescence. And so what we've shown is that pathogens use this, and symbiotic bacteria use it. So we found it in this bioluminescent bacteria, but now what we understand is that probably all, that's a big word, we haven't studied all bacteria have quorum sensing. They have to know when they're alone and when they're together so they can do these things, like kill you, that they can't do as individuals. And so now what we know, for example, is that the ability to form a biofilm, which is to cover a surface, right, to secrete virulence factors, to make toxins, all of these things where a bacteria is going to give something away to the environment. It's never going to get its own product back. All of these kinds of behaviors are controlled as a collective through quorum sensing processes. So for example, if a bacterium, a pathogen gets in you, right, it's tiny. The stupidest thing in the world a bacterium can do when it's alone is to release some toxin because your immune system evolved to see those toxins, hunt the bacteria down, and kill it. So the better idea from the bacterium's point of view is to wait, to count themselves with these small molecules, to recognize when they have the right number, that if they all release their toxins together, it will have an effect on a huge host. So now we get that if we make bacteria that can't talk or can't hear, they're not pathogenic because they need the group to act together to make pathogenesis successful. So that's where we started with this bioluminescent bacteria and then moving on to all kinds of other pathogens that you've actually heard of. And then we're also interested, okay, so what are these words that these bacteria are talking with? And so we do a lot of chemistry besides genetics in my lab. And so we started to look at, at the chemicals that just purify these molecules and figure out how these bacteria talk. And so these are just a few different species of bacteria. And this is the molecule that was the red triangle, the word, the quorum-sensing autoinducer that each one talks with. And I hope what you can see is that all of these autoinducers, right, have the same moiety on the left side, but these right-hand parts, those are just carbons, are a little bit different. So what these slight differences on the right side do is to confer exquisite species specificities to each of these languages. So each of these autoinducers really does fit like a lock and key into its own receptor and no other, just like the key to your door. And so what I mean by that is if we take the Vibrio harveyi molecule and we put it on Pseudomonas, nothing happens. Likewise, the Pseudomonas signal does nothing to Vibrio harveyi. So these molecules allow intra-species communication. These molecules allow bacteria to have private, secret conversation with their siblings, so they count their brothers and sisters and they decide when they should do something. And each one has its own language. And so once we got that far in our studies, we started to think, this is really cool. These bacteria can tell when they're alone and when they're in groups. They can talk. But the truth is, and we were doing this with this bioluminescent bacterium in a flask, but the truth is that that is not how bacteria live in nature. This is much more what the world is the wild, wild west for bacteria, right? Typically, they don't get to live by themselves. They live with hundreds or thousands of other species of bacteria. So this is a biofilm. This is actually human skin. So if we just looked at your skin under a microscope, it would look just like this. This came from your elbow, in case you're curious. And what you can see is that each one of these different shapes and sizes is a different species of bacteria in this consortium, in this biofilm. And so what we started to wonder is if it's really true that bacteria are talking to each other, if they're really counting their numbers, and these languages are so species specific, what happens when they're living in a mixed species biofilm? If you can only talk to your sisters and brothers, you're not really counting. And so we wondered, couldn't quorum sensing be more complicated than what we had been thinking about originally? And so what we decided to do is to just look a little further into these circuits, and we started looking at lots of different bacteria, and what we found is that in fact there's more than one system. So what we now know is that each species of bacteria has this system I told you. It makes a molecule, an autoinducer, and a partner receptor, and each of these are slightly different than every other species language. But then we found that all bacteria have a second system that's made up of a second enzyme that makes another hormone-like molecule, and it has its partner receptor, and all species have this same circuit. And all of that information comes into the cells. So two molecules come into the cells to tell the cells to turn on and off genes. And so we wondered, well, why do all bacteria have this second system? What's special about it that makes it different from this first system? And so we purified this molecule, and what we found out is that this is the autoinducer. And what's important about this is that this molecule is identical in every single species of bacteria. So unlike the first language, where I told you that each molecule was slightly different, which gives species specificity, this molecule is identical across all bacteria. So we think this is the universal language, or the bacterial Esperanto, that allow bacteria to talk across species. And so what we know now is that bacteria are built at least like this, which is that they have some mechanism to say me, and then they have a molecule that says other. And so the computation that we think bacteria do is the following. First, we think they just scan the environment. They say, are there any molecules out there? They're just asking, am I alone or am I in a group? So they simply scan with their receptors for molecules. And what that does is to start to set this program of behaviors. And what I should tell you is typically 200 or 300 genes are controlled by quorum sensing, right? So when a bacterium goes from acting alone to acting as part of a group, it's a big program of changes that have to take place. So first they just say, am I alone or am I in a group? And they start to turn on and off appropriate genes. And then the more complicated question that they ask is they say, is it you or is it me? And so what we've shown now is that they actually measure the ratio of these molecules, and then they change what they're doing based on whether they're winning or they're losing in any given consortium. So they test, am I in the minority or am I in the majority? And then they do very different things. I mean, you guys would know about that, right? They do very different things based on who's in the environment, right? And so we think that they're doing this very sophisticated mathematics, right, to understand their surroundings and then to turn on and off genes that are appropriate to that level. And then what I should tell you is that we've recently found it's more than self and other. So this molecule says self, this molecule says other. It does not say who the other is, but we recently found a molecule that says you're my cousin. So we know at least there's 3 words. There's a word that says me, you're my brother, you're my twin. There's a word that says you're my family, and then there's a word that says you're not, right? And of course, I will not be out of a job when I go home today. There's many more molecules to find, right? But we think that there's this complicated chemical lexicon that allows bacteria, just like we do, to gauge, who's around and then what they ought to do. And then they tailor this program of 200 genes based on this blend of chemicals that they're seeing in the environment. And so what is interesting in terms of, what I think, so this is where multicellularity came from, but I think what is interesting maybe for you guys is that if we make them defective in any of these receptors or any of these molecules, so either make them deaf or make them dumb, they're not pathogens. Right? And so where this field is going is like now that we get the bacteria talk to each other, we get that they have to understand they're in a group to be a pathogen. So the question for the scientist is, now that we've learned that, can we deliberately manipulate this conversation, the bacterial conversation, to make new kinds of therapeutics? So I assume I don't have to tell you that we have this global problem with antibiotic-resistant bugs, right? There's going to be 9 billion people on this earth pretty soon, right? So this is a major threat. Right? And there are no new antibiotics that have been developed in the past 30 or 40 years. So we're basically running out of them. And what you probably know is the way antibiotics work is that they kill bacteria. They do something. They make them so they can't make DNA. They pop their membrane. They kill bacteria. Right? And so what happens is if a bacterium fortuitously gets a mutation that makes it resistant, it and its offspring have a growth advantage. And so that's why we have this spread of antibiotic resistance. So what's happening in my field, the bacterial field, is this idea that we should stop killing these bacteria and selecting for resistant. Maybe we should be doing behavior modification, right? I mean, it works in humans. Why not do behavior modification for these bugs? And so one idea is, could we make them so they can't talk or they can't hear by antagonizing quorum sensing so that they can't count and then they don't launch these virulence attacks? And so that's exactly what we've done. We have made molecules that interfere with each of these circuits, right, just by twisting around the molecules that I showed you and like dangling different bells and whistles off them. We've made antagonists. So we know for sure if we add these synthetic molecules, we can shut off all quorum sensing in a test tube. So that looks good. And so the stage that we're at is that we now have these in animals. And I'll just show you one example, but it works no matter which of these systems you interfere with. And so the idea would be you take some pathogen, right, that could kill an animal. This year you're supposed to replace yourself with this mouse, right? So you have a pathogen that kills a mammal, right, because it has quorum sensing, and quorum sensing tells it it's inside the host and to initiate this virulence attack at high cell density, and then the animal dies. And so the question is, can we add these quorum sensing antagonists, right, so molecules that interfere and jam the receptors and get this animal to live? And the answer is yes, we can. Right? So we have several molecules that are either intraspecies or interspecies that actually save animals from bacterial infections, right? So these are now being developed into new kinds of therapeutics. These are not drugs. These are lead molecules that we have started to mess around with, and they have to be refined and made more potent and made safe, right? And so now the animal studies are continuing, but we have a lot of hope that these would actually be moved forward toward the clinic. And so that's where we are with our studies. I know it's just to give you a short talk and just talk about questions, but I hope that what you think after this short talk is that bacteria communicate with each other. They develop language, I guess, and of course they don't use real words, they use chemicals. This makes bacteria multicellular. So the computation that I showed you was invented by bacteria 4 billion years ago. And when we look at how higher eukaryotic cells do things that communicate with each other, how they use hormones or morphogens, right, and make multicellular behaviors, we realized that the molecules and the receptors and the way the information gets sent is exactly analogous to what I showed you in bacteria. So the hope from my lab is, besides just learning about bacteria, we hope that by studying these pretty simple systems, we will be able to give insight into people who study higher organisms, and also when signaling goes wrong. For example, in cancer or neurodegenerative diseases, we hope that some of these principles that bacteria figured out billions of years ago will be useful to our other colleagues, other than just studying it in bacteria to learn about bacteria. We also think that now bacteria can distinguish self from other. I showed you they have that molecule that says me, they have that molecule that says other. And of course, that's exactly what happens in your body. It's not like your kidneys get all mixed up with your heart cells every night. And that's because each of those sets of cells use chemicals to drive their fates and to tell them what jobs to carry out. Again, we believe bacteria invented that, and by studying bacteria, we're going to actually understand a lot about higher organisms and how this kind of information goes around in higher organisms as well. And then, of course, the very practical part is that now everyone wants everyone. Lots of people in my field want to develop strategies to impede quorum sensing in the harmful bacteria, but also to go back to my very beginning, right? Most bacteria in this world are really good for us, or they do things that we want. And so the other strategies that we're working on now is to make pro-quorum sensing molecules. So could we make beneficial bacteria talk better to be able to get them to do things we want? And so, for example, right, no plant would grow if it didn't have these symbiotic bacteria at its roots giving it nitrogen. That's controlled by quorum sensing, right? So can we use this to get crops to grow reliably with fewer inputs, or in desiccation, places that get desiccated? Can we are going to use microbes to make biofuels, right? That's clear, right? Could we use quorum sensing to get those bacteria to really be machines at making either biofuels, or we already use bacteria to make hundreds of different kinds of medicines, right? We use them as little powerhouses. And so maybe if we could exploit quorum sensing in the naturally good bacteria, or the bacteria that we're trying to get to do something we want, that's also another avenue. So this is being played both ways in my field now, which is to stop bad bacteria from talking and to make the conversation of good bacteria better, right? And then there's also so that's just for all kinds of biological research, but there's also other kinds of industrial research. Is like people are trying to hang these pro and anti-quorum sensing molecules off plastics. Like if you're going to get a catheter, you're going to get an infection, could you make that catheter be anti-quorum sensing? People are painting pipes, where you have like industrial plants and the pipes get all clogged up. That's a bacterial biofilm. So could you put these in paints and stop that from happening, right? Put these in stents, put these in, knees, all kinds of different artificial materials, right? So there's all of this biological and industrial and agricultural, and I think also the energy field as well, uses to learn now that bacteria talk, right? And then depending on what you're trying to do, there's a lot of ponies in the race to get them to either stop doing it or do it better. And I guess what I would just say, and it kind of echoes what Rush Holt said, is that what you have to remember about this field, right? So there's this very practical medical application. We get now that bacteria couldn't kill us if they didn't have quorum sensing. So that's being worked hard. But now we're starting to see that there's all these other beneficial things, like in the agriculture and energy and food environment, right? And this all started with a harmless bioluminescent bacteria, right? And 20 years ago, the NIH and the NSF took a flyer on me, right, to say, that's kind of cool, right? And all I was studying was why this bioluminescence turned on together, right? And I wish that I was so smart to have thought, oh, we're going to cure all these diseases, we're going to make food for everybody, of course this is more than Vibrio harveyi. We thought we were working on something fundamental, like how does information get relayed into the cell? We thought it would be more than one crazy bacterium from the ocean, but we never thought it was going to be all bacteria, that there would be all these molecules involved, right? And that there would be all of these very practical health and environmental related issues, right? But a long time ago, when there was no such thing as this field, there's just me, right? Somebody took a chance, and now hundreds and hundreds and hundreds of labs around the world work on this, right? And so I just would make a plea that that is what, when the NIH and the NSF and the ONR are working at their best, right? They are taking flyers on young people that are studying somewhat crazy things, but you just don't know where the possibilities are going to go. And so I'm not the young person anymore, so I want to just finish by showing you my gang, right? So this is my lab at Princeton, and of course what you also know, right, is that the science in this country is always done by 20 to 30-year-olds. People like me type, right, and get to come to the Rayburn building. But you know, that everything that I told you, every idea, every molecule was discovered by somebody's hands and someone's brain who's in that picture. So that's my gang, and they had all of these ideas, and they're all going to go off to do something that has to do with science, right? And I think that it's a really fun enterprise to get to work with. And I think the other thing that maybe you guys understand, but most people don't understand, is everybody thinks that science gets done by boring old white men. But the truth is that it gets done by 20-something-year-old kids. And it's really a rush for me to actually be involved in that, because it's like, you know, being with this amazing energy every single day. And so I want to thank you for the privilege of getting to have this to be my job, the zookeeper. Anyway, but thank you for listening to that. I'm happy to answer questions about bacteria.
I have a question.
Yeah.
Oh, sorry. I mean, you mentioned that you were basically attempting to make the bacteria basically dead both deaf and dumb. Could that possibly lead to them to just attack without caution, without regard whether they're alone or?
Yeah. So right. So I don't know if everybody can hear you. Yeah. So the question is, if you really make them so they can't communicate, right? Would they just turn their stuff on anyway? And so the answer is they won't, right? So if you make them so they can't count, they think they're alone and that they shouldn't. I always promote that they shouldn't turn on these virulence factors, right? So we know they just, if you make them miscount, they don't turn on virulence factors, right? So, because they have been programmed over 4 billion years to understand that virulence factors are incidental if there's not lots of bacteria together.
I'm guessing that if you're attacking some sort of harmful bacteria, you're going to try and communicate with some cousins because there's different strands. Do any of those cousins end up being beneficial?
Right, so these are all really great questions, right? So the question is, so he's talking about specificity, right? So when physicians think about this, there's many different strategies. One is if the physician knows what you have, like if you have cystic fibrosis, you are going to die of a Pseudomonad infection. So you want a drug that treats Pseudomonas. So in that case, you want to go for that very species, try to interfere with the very species-specific system and not any of the others. But oftentimes, if you go to the hospital and you're sick, your physician has no clue what you have. So you get treated by broad-spectrum antibiotics while they narrow down trying to figure out what you really have. In that case, you'd like to go for the interspecies, the universal one, right? So you would try to not be specific. And then there's all these things in between, like, for example, E. coli, which there can be bad E. coli if you get E. coli 0157, but you depend on E. coli in your gut, good E. coli, to digest your food, right? So there's all these different ways to play it. And so the real answer to your question is that we have evidence that we can actually make molecules that are either very specific or less so or very unspecific. So that's good news in terms of just thinking about how we would deploy these different strategies. But so the answer is yes, right? But also what's really interesting about this, so let's just say this, I think a deeper question that you're sort of asking me is, like, let's say we went against this interspecies communication, and I could make some fantasy drug that shuts down quorum sensing in everybody. And you go to the hospital, you're really sick, you get that drug. What about all the bacteria, the good bacteria in you? They're also going to be goofy and unable to communicate. That actually turns out to be okay, because if you think about every antibiotic you've ever taken, it does not just kill the bad bacteria, right? That's why people get a stomach ache, women get a yeast infection, because when you take an antibiotic, you have these trillions of bacterial cells, 10 to the 14th bacterial cells in your gut, right, that get mostly get killed. And so then your flora gets all goofy and maybe you feel sick. What is the miracle of bacteria is that you have what are called persister cells. And so these are little pockets of these bacterial cells that aren't harmed by these drugs. And so after you finish taking a course of a regular antibiotic, they're there and they repopulate, right? And that's why you can take antibiotics, right? And you're not just dead, right? And so what is amazing, right? And so we're counting. So there's 70 years of research showing doing something much more harmful, which is really reducing the numbers with traditional antibiotics, actually it doesn't matter that you've actually treated all the other bacteria. And so we're banking on that with these sort of non-specific quorum-sensing drugs. And of course we want to make these specific ones for particular therapeutics as well. But what is so amazing is when antibiotics became industrialized, after like World War II, right? Nobody knew these bacteria were living in you or on you. It is amazing they ever worked, right? Right? And so, right, that's what's so cool is that this host microflora, which I would like to tell you, you inherit from your mother and father, right? As soon as you come out of the birth canal and all your slopping spit, you get this microflora when you're a few months old. You have it your whole life. And the reason we're mostly not sick is because it is so hard to perturb that consortium. And so even when you take an antibiotic that kills 90% of the bugs, like in your gut, right? These little protected cells, and we don't understand who those persisters are, but they're there, and then they just grow back. And so the point is, and of course, this remains to be seen. I mean, we don't have this drug yet. But using what we know about traditional antibiotics for this last 70 years, we're pretty confident, and the animals at least show this, right? That everybody is like either talking or not talking, right? All the wrong words, right? And then once you take away the molecule, in fact, at least the population is still there, and then they just start chit-chatting again like they're supposed to. So we're pretty confident about that part. If you ask me the parts I'm not confident about, I'll tell you, but I'm not telling them until you ask. Yeah. Yeah.
So thanks for a great talk. If we're successful in creating mechanisms for suppressing this communication, then it doesn't seem that it wouldn't be that hard to create mechanisms to enhance the communication of bad genes, and that could have some biosecurity implications.
Okay, so all of this, yeah, so right. So if we can make pro-quorum sensing molecules for beneficial bacteria, you can make pro-quorum sensing molecules for harmful bacteria. And so one thing that it's so sure, right? Like, and so that's totally true. One thing that is interesting about the pro-quorum sensing strategy in the sense of pathogens, right, is that if you turn them on too early, right? So you have a few bugs, and you're a terrorist or whatever, makes a pro-quorum sensing molecule, and so then now I release all my virulence factors, but there's too few of me. In fact, that doesn't work either, right? The bacteria have to count to the right number so that the factors there's enough there, right, to do harm to the host, right? So the truth is that in the sense of virulence, anti-quorum sensing molecules or pro-quorum sensing molecules that get the guys to show their stuff when they're too few and then the immune system gets rid of them, both of those can kind of work, right, as a strategy. But I still want to say that, of course, what I do is basically glorified cooking. And if somebody could figure out that I could give that molecule pro-quorum sensing at the right time, right, and get these bacteria to turn on virulence, yeah, it would have biosecurity problems, right? Like any cloning, like any, and that, and of course, we're not suggesting that we're going to release this tomorrow, but I think those things have to be thought about. But I also think that the real threat is that there's 9 billion of us in 50 years and we don't have any antibiotics, right? I think that's a much bigger threat. And, and I assume that you guys are gonna, we're gonna do science to the best of our ability. We have a rigorous standard of truth in my lab, and then I assume that people who think about the ramifications of that are gonna think about that with us, right? And help us think through that part that we're not expert in. Mm-hmm.
You said you've identified some of the genes that make these quorum sensing molecules. Have you been able to use those genes in different species to produce those molecules along the lines of, is it these quorum sensing molecules that make individual bacteria individual species? Or because there seems to be uniform
So
body types?
Yeah, so it's easy, and this goes to your question. This is like glorified cooking. We can move these genes around. It's actually pretty easy. So we can make, an E. coli that makes the Vibrio harveyi molecule. We can do all of those kinds of things. And then, if you put the whole circuit into E. coli, they will act like Vibrio harveyi, right? And so what people think, and this is work of Rich Novick's, is that if you look, and this goes back to what you're saying, if you look at species that are very closely related to each other, right, like different Staph aureus species, what you can see is that this is about speciation. So you can look through the evolution by looking at the genomes and see that the molecules and the receptors are changing hand in hand. You know, because if you change your molecule, then you can't talk. You know, so they're changing, and you can just see through species that they're like cousins, right? How they're like at first you have a very strict specificity, then it gets loose, and like the receptor can respond to a couple of molecules, then the next guy down responds to a new molecule. And so people are starting to think that the ability to communicate has something to do with speciation, right? Because then what happens is that they tailor the genes at the bottom of the cascade based on what's happening outside. And so that is one idea for what it means to be a particular species of bacterium versus another. I think that's a cool idea. Of course I do. I'm brainwashed. I think quorum sensing runs everything. Yeah.
Has there been any evidence that individual bacteria are able to use this quorum sensing technique to communicate with other microscopic organisms or multicellular organisms?
So that's totally where this field is going. So first of all, this idea of this pro and anti-quorum sensing strategy. So first of all, yes. So some species have slop. Some systems have slop, you know, so they can talk across species boundaries. No one has found so what I showed you today, there's interspecies, intergenera. I didn't show you that. Intraspecies, intergenera, you know, within the family, and then interspecies communication. Is there interkingdom communication? So now lots of people are working on bacteria that are in the gut, right? And asking, are those bacteria driving what you do? And likewise, are your cells telling the bacteria what they ought to be doing? And the answer is becoming yes. But it's very early days. So to give, this is 20 years. We didn't know bacteria talked to each other 20 years ago, right? So now this idea of do they talk to the eukaryotes, right? That's what's on the horizon. And so people are studying that hard, right? In terms of like things that bacterial world, that's totally cool. So, you know, like I have this idea that I and these couple 22-year-olds are going to make some chemical that interferes with quorum sensing. Well, then we had the idea, the bacteria have had 4 billion years to do that, right? And they're out there, you know, fighting it out in all these, you know, restrictive niches. Maybe they already discovered, you know, made that, and anything that's out there in the natural world is going to be a lot better than anything those guys can make or me, right? And so we went out and you started mining for, you know, looking at, you know, dirt and plants and stuff, and sure enough, the bacteria out there, they eat each other's autoinducers. You know, so if I eat yours, you can't count, right? They actually make antagonists, right? Just like the molecules we're making. They eavesdrop, so they free ride, you know, and then like if you're making an autoinducer, I respond to it, but I don't participate. They actually make they make these antagonists, and then other bacteria make like a molecule that for us, it's our autoinducer, but it's actually a poison for you. So there's this sort of double information in it, which is we undercount and you get killed. And so they're doing all of these amazing things, right? And so now a huge part of this field is to just go out and ask, you know, what solutions are out there? Because if they exist out there, that means they're good ones, right? Because they've persisted over 4 billion years, right? And so all of those things, like you know, the molecules I showed you with the rings and the chains, there's some guys that talk with that, and the guy next to it has an enzyme that cuts that chain off the ring. You know, so this guy's trying to talk, and the guy next to it in the dirt is making it dead and making it mute, right? And so all of those shenanigans are going on out there, right? And so I think that's a very profitable place for people to start looking for applying the natural ones, you know, with bells and whistles to industrial or, you know, medical uses, right? But that's pretty new because, you know, first we, you know, the field found this, and then we're like, we're trying to interfere with it. They probably already, you know, right? I mean, you see how it's going. But that's a pretty fun avenue that we're working on right now.
One question I have is, do you think that these sorts of therapeutic techniques will fall into the same trap as antibiotics?
Yes, absolutely. Oh, yeah, sorry, yeah. Yes, I need a job. Yes. So your question is, are they going to get resistant? Yeah, so they will, right? So these are bacteria, and bacteria do evolution on a 20-minute time scale, right? And so I feel like that if we can make this pro or anti-quorum sensing strategy, it's just to buy time for someone to find out the next behavior, right, to target, and other behaviors are being targeted. One thing that is interesting that we, meaning in my field, think about in terms of this idea of modifying their behavior versus killing them, right? So I already said this. When you give a traditional antibiotic, right, the bacteria get killed. So if some individual in that population has a mutation that makes it resistant, it's not being killed. So it and its offspring are the ones that grow. So there's this immediate growth advantage. What happens here, and we've done some experiments, is if you have a guy that you would say is resistant to this molecule, right? So I give this drug, right, and this bacterium doesn't perceive it, right? What happens is that so remember, it's in a population, so it's counting, and this goes back to your question. So it's counting, right? And it decides, oh, there's lots of us. I'm going to turn on my quorum sensing cascade. But everybody else, right, is being dealt with the drug. They're miscounting. And so this guy, so what happens, at least initially, is the guy who is resistant, by the traditional is resistant, it turns on its quorum sensing and it starts pumping out these hundreds of genes, and everybody else isn't participating. So it actually has a growth disadvantage because it's working so hard, but it doesn't reap the collective benefit. So at least the traditional way that bacteria get resistant, which is to just avoid the drug, that is not it. They're going to get resistant, and what we're trying to do now that we have these drugs, is we're just selecting for resistant mutants that live, and we just sequence their genomes and say, what's wrong with you? But so the only fantasy is, because you're not outright killing and giving this advantage for fast growers, is that you get a longer shelf life, a functional life, out of therapies that are dealing with bacterial behaviors, so quorum sensing or other ones, right? And so the fantasy is that you just get a longer time, right, to work toward the next thing, but they're definitely going to get resistant. We're not so dumb as the first time that this, we can learn. Yeah.
So an advantage, therapeutic-wise, at least for antibiotics, is they're cheap, they're stable. How feasible is this as, even with the experimental therapy? Easy to do, is it expensive?
So it's expensive. Right now, it's, so nobody has made these molecules on the, 55-gallon drum level. So it's very expensive to be making these right now. The molecules are very simple. The antagonists we have are very simple. So theoretically, actually, if we chose to they're not expensive molecules. They're not hard syntheses. Antibiotics, right? Yeah, they're cheap and whatever, but they don't work, right? And so that's going to be an advantage, right? But you're right. No, we're not even close to that level where you would start to think about scaling up at the level you're talking about, because we haven't increased the potency enough and the sort of built into the molecules all the safety parts yet, and so we're not even close to that. But at least sort of looking at it from far away, the molecules are really simple, so that's really good because they're like two-step syntheses, and so one could imagine easily scaling them up. At least that's the fantasy. And you're right, they're only going to get used because they have to be used, right? Because you can get Riff for pennies or whatever, right? And so, yeah. Mm-hmm.
You said that the body recovers from the onslaught of antibiotics because of persistent cells that can do the pathogens not have to deal with?
So what's cool, right? So no, they don't. So what's amazing about this, so what we now understand is that your immune system is working hand in hand with your microbiome, your commensal microbes, to keep pathogens out. Right? So what happens is the only reasons pathogens get to, and it works most of the time. I mean, you guys just ate all that stuff. It's covered in bacteria. You will not be sick, I think, this afternoon, right? And so what happens is that pathogens, the reasons pathogens can get a toehold is because they have some bell or whistle, an adhesin, a toxin, some kind of poison that allows them to stay. And so if they can't launch that, right, which is controlled by quorum sensing, in fact, the immune system hunts them down, sees them as foreign. Your microflora helps just take up all the space and gets rid of them. And so that's what's amazing, is that pathogens have to have the things that make us sick in order to stay, right? And so those are controlled by quorum sensing. So there is no such thing yet as a persister that's a real pathogen.
Nothing that can hide from...
There probably are things like that. And then there's also this whole thing, I don't know if you guys have heard of this bacterium C. difficile. Have you heard of that? Yeah. So we have all these things that we now call pathobionts, meaning they're sort of symbionts and they're sort of pathogens. So C. diff is this bug that lives in you all the time at a very low level. And all your other bacteria keep it in check. But then if people take antibiotics and these ratios get screwed up, then it's a normal bug, but when it gets too high, it actually makes a toxin and people get really, really sick from this after they've taken antibiotics, right? And so that's like this idea that these bacteria are only symbionts when they're in the right place, right? If you get a wound to your gut and you spill all those good E. coli into your blood, you're dead, right? You know, and so there's this whole environment where you keep these bacteria at the right number in the right place, but they could, and so they're not pathogens to you, they're required, but that's because they're where they're supposed to be. And so it's sort of like this goes along with this speciation, like what's a beneficial, what's a bad bacterium, it depends on where they are, right? So, but at least in principle, what you would consider a pathogen, so I mean by a bug that never was there before, you know, you would get food poisoning or you get some infection, right? They can't stay if they don't launch these attacks. That's how they get there in the first place, get to be there in the first place. Mm-hmm.
Do you think it's possible that, I know you mentioned finding solutions that already exist in nature, but most of those solutions exist in nature because they have a measured level of success, and if they become too successful, generally the environment gets destroyed and they end up back themselves. Wouldn't it be more successful if you specifically target one species using the quorum system and something that's significantly more harmful to that one species that doesn't affect anything else?
No, I think that idea, I think that is the ideal. If you could make an extremely selective, like, I don't know, pick Pseudomonas again, right, this pathogen that's in the hospital. If you could make an extremely selective molecule for each, you know, bad bacteria, you know, that would be cool. Can we do it? You know, these molecules aren't that different, right? So trying to encode that kind of exquisite specificity might be hard. I don't know the answer, but absolutely, we would love to make, you know, like your doctor, you're not taping this part, your doctor looks and he goes across and says, I give this molecule, right? But remember, so that's a great idea. The problem is when you really think about infections, at least in the present, most of the time the physician doesn't know what you have, right? So there's always going to be a role, I think, for these broad spectrum and narrow spectrum drugs, right? And depending on what illness you have, the cocktail of those drugs you get is going to change, right? You know, but we fantasize about making, you know, like really like cocktails, you know, of 3, you know, and stuff like that. But I think that there's still this place where you want a very generic quorum sensing or, you know, antibiotic, right? Because you don't know what you have. And, you know, with genomics and PCR, that may get really fast, you know, where doctors actually do know what you have, you know, because now they still grow the bacteria, you know what I mean? And so it takes a few days. And so, of course, you know, so I'm thinking right now, but it could be that in the future, you know, they just take a little sample and 20 minutes later they have the DNA of the bacterium. And then, you know, the fantasy that you get this sort of precision medicine, right, would be cool. And that, I think, is where the kind of strategies you're talking about would be amazing if we could have, you know, a set of molecules for, or even for just groups of bugs, right? That would be cool. Yeah. Mm-hmm.
And you've been talking about a lot of how doctors don't know what specific bacteria we have. Looking at the specialized quorum molecules, have we been able to test for the presence of those specialized molecules?
Yeah, so people, right. Yes, so people are trying to make dipsticks now, like, right? Like this would be a part of not just therapy, but diagnostics, both for medical and also in the environment, you know, like you have pollutants or you have some kind of contamination, right? So trying to understand what is the lexicon, right? And can you make sensors, you know, that are very specific, right? Just by attaching the receptor proteins, that's in business now. You know, we know those few molecules, right? But again, it's getting easier now that we know that quorum sensing exists. It's getting standardized. You find, you know, your pet bacterium that lives wherever, you can pretty quickly now at least find what the molecules, at least these few that we know about, are. And I think that kind of thing where you're making like sensors for whatever, right, is really going to be a good industrial thing. You don't know, for water testing, food testing, right? Like, you know, E. coli 0157 makes this, you know, so you test the meat. I mean, I think that people have their eye on that prize for sure, right? Much more like the engineers, right, who make those kinds of gizmos, you know, are working hand in hand with the biologists to do that. I think that's super fun, and you can think of a gazillion applications for those kinds of things. Mm-hmm. Thank you, you guys. It's really nice of you to take time.